everyone, I'm Kavita from MSc Botany. I'm going to present on the topic polyembryony. Polyembryony has been defined as the occurrence of more than one embryo in a seed. They may become arrested at very early stages or may degenerate during the course of seed development. Therefore, the number of embryos formed is generally higher than the number of seedlings formed by a polyembryonic seed. Of the 75 woody species investigated, around 14 species belong to 12 different families exhibited polyembryony. The first case of polyembryony was reported in certain orange seeds by Anton van Leeuwenhoek in the year of 1719. Polyembryony is quite common among the conifers in gymnosperms and dicotyledons and in monocotyledons in the angiosperms. Polyembryony in angiosperms may arise by cleavage of proembryo, formation of embryos by the cells of embryo sac other than the egg, development of more than one embryo sac within the same ovule, or activation of some sporophytic cells of the ovule. Next, occurrence of polyembryony. Many theories have been advanced to explain the occurrence of polyembryony, but none is sufficiently validated. In different theories, polyembryony has been described to hybridization, necrohormones, the effect of recessive genes, etc. Haberland in the year of 1921 proposed the necrohormone theory. He regarded the degenerating cells of the nucellus as source of stimulus for the adjacent cells to divide and form adventive embryos. Haberland attempted to induce adventive polyembryony in Enothera by damaging cells by pricking the ovules with a fine needle and by gently squeezing the ovary. In one ovule, he got 2 embryos which he considered to be of nucellar origin. Attempts of subsequent workers to induce adventive polyembryony using Haberland's method yielded only negative results. And the another theory is hybridization theory, also called as recessive gene theory. According to this theory, the occurrence of multiple embryo is due to hybridization. According to Capet in the year of 1935, the polyembryony is a recessive heredity character formed by a group of multiple genes. The recombination of genes takes place during the present hybridization, forming a single unit that gives rise to the multiple embryos. One of the example is polyembryony that is found in mango that is caused by one or more recessive genes. Here we can find a few Indian varieties of mangoes such as Bombay, Fazli and Langra. They are polyembryonic varieties. Spontaneous polyembryony has been subdivided into 2 categories. One is true polyembryony and the other is false polyembryony. In the true polyembryony, 2 or more embryos arising in the same embryo sac from zygote or embryo. It can be found in manda or synergid, sagittaria, antipodal cells in ulmus or nucellus or integument in the citrus varieties. This type gives the result of development of aposporic embryo sacs. Later on, Gustafson proposed that the term false polyembryony should be restricted to those cases only in which 2 or more nucellae, each with its own embryo sac, fuse at the early stage. He also proposed that true polyembryony is restricted to embryos derived from one or several embryo sacs found in the same nucellus. One of the true polyembryony types is cleavage polyembryony. Cleavage and proliferation of zygote or its derivatives leading to the establishment of separate embryonal primordia is widespread among gymnosperms. In angiosperms, this feature is less frequent. Among angiosperms, cleavage polyembryony is quite common in orchids. In Eulophia epidendria, Swami in 1943, he recorded 3 different modes of supernumerary embryo formation. First, the zygote divides irregularly to form a mass of cells, of which those lying towards the chalacal end grow simultaneously and give rise to many embryos. The proembryo form small buds or outgrowth which may themselves function as embryos. The filamentous embryo become branched and each branch form an embryo. While the cleavage embryony arising during the seed development that is found in many orchids, the formation of plural embryos during seed germination is known only in Vanda. In this genus, the apical promeristem of the embryo divides into a number of primordia, around 3 to 9, each of which further organizes into an embryo. And the another type is adventive polyembryony. The embryo formed from the cells of the nucellus which penetrate into an embryo sac. Some of the examples are citrus, eugenia, and mangifera. Some of the embryos may be produced from other parts of the embryo sac. The most common source of additional embryo is the synergid. Depending on whether it arises from fertilized synergid or unfertilized synergid, the embryo may be diploid or haploid. In the Aristolochia, Bracteata, Poa alpina, and Sagittaria gramineae, besides the egg and the pollas, one or both the synergids may get fertilized. This can be brought about either by the entry of more than one pollen tube into the embryo sac or by the presence of additional sperms in the same pollen tube. In such a situation, the zygotic as well as the synergid embryos are diploid. Embryos arising from unfertilized synergid are known in Agemon mexicana and Phaseolus vulgaris. In such cases, the zygotic embryo can be distinguished from the synergid embryo that can be haploid by its diploid nature. Formation of embryos from Paspalum scrobiculatum, Ulmus americana, and Ulmus glabra. The antipodal cells may divide a few times to form proembryolic structures. However, they fail to grow into an adult embryo, and there is a suggestion of antipodals forming germinal embryos. Most of the reports concerning endosperm cells forming embryos are doubtful. However, Brachyria scitigera, an Apomis species, it is one of the examples where endosperm has been reported to produce triploid embryos and seedlings. Here, the picture depicts the citrus ovule in the young seed, showing normal and nucellar, that is adventitious embryos. And the picture in the right side depicts the formation of synergid embryo in the plant Lilium. And in this picture, the left one is the development of embryo from the antipodal cells, and the right one is the embryo sac of Ulmus glabra, showing zygotic and antipodal embryo. Suspensor polyembryony. It is the common feature in the genus Exocarpus, a member of Santalaceae. As many as 6 embryos may develop simultaneously in an ovule by the proliferation of the suspensor cells. Eventually, however, Only one of them takes the lead and reaches maturity. Rarely, two germinal embryos may be formed in a seed. In the zygophyllum fabaco, the suspensor embryos may develop up to the heart-shaped stage. And the next one is nucellar polyembryony. Apomictic procedure is involved where many tenderly consequent embryos stand from the nucleus and surround the emerging zygotic embryo. Nucellar polyembryony occurs in the crassinucellate ovules, example citrus, Opuntia sarcoca. Spontaneous polyembryony classified into two types on the basis of genetic variation. One is gametophytic polyembryony, that is arising from any gametic cells of embryo sac after or without fertilization. And the second one is sporophytic, that is arising from the zygote, proembryo or initial sporophytic cells of the ovule. It can be nucellus or integuments. In some plants, the embryos develop from the endosperm. However, the embryo develops normally from the egg embedded in the cellular endosperm and does not develop from the endosperm proper. And the origin of embryos from the cell outside the embryo sac. The embryos also develop from the cells of the nucellus and arranged integument. Such development of embryos has been reported in citrus, Syzygium and Mangifera. The embryos, although initiated outside the embryo sac, later on come inside and are nourished by the endosperm. Importance of polyembryony. This phenomenon plays an important role in plant breeding and horticulture. Nucellus adventitive polyembryony is of greater value in horticulture, where the nucellar seedling of citrus has been proved to be better clones of arched stock than cuttings. On the other hand, the nucellar embryo are supposed to be free from disease, and the nucellar seedlings regenerate the vigor that is generally lost after continued cutting propagation. The adventive polyembryony is much useful in the propagation of the fruit trees such as citrus and mango. The application of adventive embryony is also important for providing genetically uniform seedlings in fruit trees. The haploids can be used for the development of homogeneous diploids, which are of high in the value. Due to the practical value of haploids in plant breeding, the methods have been recognized for the artificial production of these embryos from the eggs and synergids. Following are some of the textbook references for the polyembryony. Thank you.